Hello, a warm welcome from SGT University. I am Dr. Ashok Yadav, Faculty of Agriculture Sciences. Today we are learning about the breeding of mango. Mango Mangifera indica is the leading crop of India and considered to be the king of fruits. Mango occupies 22% of the total under fruits comprising of 1.2% million hectares with a total production of 11 million tons. Mango is an important cash crop in the tropics and subtropics, determining the yield gap of mango and production constraints can potentially promote the sustainable development of mango industry. The long gestation period for evaluating the hybrids and the long testing time for the selecting hybrids for commercialization has been essential in mango breeding and to produce pure breeding progeny superior in the many respect to the parental types. Hybridization in mango during the 20th century. Planted hybridization between carefully selected parents has become dominant in the breeding of self-pollinated species. The objective of hybridization is to be combine desirable genes found in two or pedigree breeding starts with the crossing of two genotypes, each of which has one or more desirable characters lacked by the others. If the two original parents do not provide all of the desired characters, a third parent can be included by the crossing it to one of the third progeny of the first generation. In the pedigree method, superior types are selected in the successive generation and record a maintain of parent-progeny relationship. The bulk population method of breeding differ from the pedigree method primarily in the handling of generation following hybridization. The F2 generation is shown at the normal commercial planting rates in the large plots. At maturity, the crop is harvested in mass and the seeds are used to establish the next generation in a similar plots. No record of ancestry is kept. During the period of bulk propagation, natural selection tends to eliminate plants having poor survival value. Two types of artificial selection also are often applied. Number one is the destruction of the plant, they carry undesirable major genes, and number two, mass techniques such as the hybrid harvesting when only parts of the seeds are mature to select for the early maturing plants or the use of screens to select for the increased seed size. They are the mass selection. In mass selection, seeds are collected from the usually a few dozen to a few hundred desirable appearing individuals in a population, and the next generation is shown from the stock of mixed seeds. The procedure, sometimes referred to a phenotypic selection, is based on how each individual looks. Mass selection has been used widely to improve old land varieties that have been passed down from one generation of farmers to the next over long periods. Self-pollinated crop. Mass selection. In mass selection, seeds are collected from the usually a few dozen to a few hundred desirable appearing individuals in a population, and the next generation is shown from the stock of mixed seeds. The procedure, sometimes referred to a phenotypic selection, is based on how each individual looks. Mass selection has been used widely to improve old land varieties that have been passed down from one generation of farmers to the next over long periods. Cross-pollinated crop. The cross-pollinated crop, there are generally three types. That is, the number one is pure line selection. Pure line selection generally involves the three or more less distinct steps. Number one, numerous superior appearing plants are selected from genetically variable population. Number two is the progeny of the individual plant selection are grown in the evaluation by simple observation frequency over a period of several years. Number three, when the selection can no longer be made on the basis of observation alone, extensive trials are undertaken involving careful measurement to determine whether the remaining selections are superior in yielding ability and other aspects of performance. Mass pedigree methods. in cross-pollinated crops. In this method of breeding, the best individuals with desired characters are selected on the basis of phenotypic performance in a source's population. Open pollinated seeds of the selected individual's plant are divided into two halves. Second year replicated progenies row trial is conducted using one set or half seeds from the each plants. Number two is the inbreeding methods. In cross-pollinated crops, the mating of individuals more closely related than individuals mating at the random is known as the inbreeding. The lines produced by the continued inbreeding are known as the inbreed lines. Self-fertilization is the most intense from the inbreeding. In plant breeding, nearly homozygous lines are produced by the continued self-fertilization accompanied by selection for 5 to 6 generations. This can be used as the method of breeding only in those crops which do not show any loss of vigor due to inbreeding. Flower biology of the mango flowering. The mango flowers are simple, open and small in size. They are visited and pollinated by the many different kinds of insects. Mango pollinators including different kinds of apes, flies, stingless bees and oven ants. Flies and stingless bees are thought to be the main pollinators of the mangoes as they visit the flowers in the large numbers and disperse between the different trees. As mango pollinators are wild insects, the main things that support them are patches of the natural habitat and wildflowers to the visit when the mango trees are not in flowers. Farmers growing mangoes need to be aware of the importance of the pollinators for the mango production. The mango inflorescence is primarily terminal, although axillary or multiple panicles may also arise from the axillary buds. Both perfect hermaphrodite and staminate male flowers occur in the same inflorescence. The total number of flowers in a panicle may vary from 1000 to 6000 depending upon the cultivars. Initial fruit set in mango is directly related to the proportion of the perfect flowers, although the final fruit set does not necessarily depend on the ratio. It appears that the proportion of the perfect flowers in a cultivar becomes critical from the optimum fruit set only when the proportion drops to the 1%. Anthesis of flowers. Flower begins to open early in the morning and anthesis has generally been completed by noon. The greatest number of the flowers open between 9 to 10 am. Although the receptive of the stigma continues from 72 hours after anthesis, it is the most receptive during the first 6 hours. However, there are reports that the stigma can become receptive even before anthesis have occurred. The minimum time required for the pollen grains to germinate is 1.5 hours. Pollination. Mango is the cross-pollinated which is carried out by the insect which has the commonly housefly, honeybees and thrips and possibly by the other insect although to the lesser extent. Pollination by the wind and gravity has been suggested to occur in the mango. In nature, more than 50% of flowers do not receive any pollen. Some workers had suggested that self-pollination in certain cultivars can also occur quite frequently. The optimum temperature for the normal meiosis is between 15 to 30 degrees centigrade with 70 to 85% viability. In addition to this natural fruit decline, young developing fruit primordia disappear before maturity, suggesting that the senescence and shedding are independent phenomena caused by the hormone imbalance. Ethylene stimulation, cellulose synthesis, and its effect on the cell walls by the upregulating genes that express cellulose and pectinase. These genes will lase interlayer cells and cell wall and form the separate layers. Eventually, decline of flowers and fruits. Mango usually fall from the stem initially not because of any insect or other problems. A tree can't support all the heavy fruits. Only a certain percentage of the tree can mature into the complete fruits. The fruit set ratio is low 0.1% and the young fruit is greatly reduced 99.9% causes huge losses to the mango growers. discarding some mango fruits as a normal tree thinning process with the goal of properly utilizing the available ingredients. Immature foods scramble to buy available food and water. Although the substantial reduction in the mango fruit is due to the many factors such as lack of pollination, failure of fertilization, ovule and germ degeneration, and low sex ratio. The strongest will survive. Short stemmed apple mango variety, especially clustered mango, will be harvested close to each other. Early morning seedlings, especially reducing the fruit cluster, will help prevent the decline after picking fruits. They stimulate ethylene and cause more premature crops. So today we covered breeding of mango. Next time we meet you, we will be exploring about breeding of banana. Keep learning, keep growing. See you next time. Thank you.